Hello students, today we'll see the next methods. In the previous lecture we have seen about the Dumas method, Biuret method and infrared spectroscopic method for the determination of the proteins from the given sample. Now today we will see other methods like Lowry method and second one is the dye binding method. So for the dye binding there are two methods. First one is the anionic dye binding method and second one is the Bradford dye binding method. We'll see one by one. So the first one is the Lowry method. So the Lowry method combines the Biuret reaction with the reduction of the Folin Ciocalteau phenol reagent, that is which consisting of the phosphomolybdic acid and phosphotungstic acid, by the tyrosine and tryptophan residues which are present in the proteins. So the bluish color develop, which is read at 750 nanometer, which is having high sensitivity for the low protein concentration, or whenever we have read the absorbance at 500 nanometer where the low sensitivity for the high protein concentration is occurred. So the original procedure has been modified by Miller and Hatry to improve the linearity of the color responses to the protein concentration. So these are the amino acid, tyrosine and tryptophan side chains which are taking part in the formation of the particular complex, or we can say that there is a formation of chromophores which shows some kind of color. Now the procedure. So the following is the procedure which is based on the modified procedure of Hatry. So the first one, we have to analyze the protein which are diluted to the appropriate range. So we have to dilute the given sample having concentration 20 to 100 microgram. Okay, then we have to add potassium sodium tartrate and sodium carbonate solution into it, and after cooling we have to incubate it at room temperature for 10 minutes. Then copper sulfate and sodium potassium tartrate and NaOH solution is added after cooling and incubated at room temperature for 10 minutes. Okay, so first of all what we have to do, we have first take the protein solution, into that we have add sodium potassium tartrate and sodium carbonate solution, and then we have to cool it and then incubate it. After that to the same we have added copper sulfate NaOH solution, and after cooling and incubated at room temperature for 10 minutes. Then we have add freshly prepared Folin reagent to the same, and then reaction mixture is mixed and incubated at 50 degrees Celsius for 10 minutes. Then there is a development of the color. Then we have to read the absorbance of the color at 650 nanometer. A standard curve of DSA is carefully constructed for estimating the protein concentration of the unknown. Okay, so as we have know that we should have to plot a calibration curve, or we can say the standard curve, by using the BSA, and then we have to find out the concentration of the unknown. Then what are the applications? So because of its simplicity and sensitivity, the Lowry method has been widely used in the protein biochemistry. However, it is not been widely used to determine the proteins present in the various kinds of food samples without extracting. Then what are the advantages? This method is very sensitive, so about 50 to 100 times more sensitive than the Biuret method. So from that Biuret method we have used the Lowry method. So it is the combination of Biuret method. Then 10 to 20 times more sensitive than the UV absorption method, which is occurred at a 280 nanometer. Then similar sensitivity as a Nestlerization, however more convenient than the Nestlerization. So this Nestlerization, it is nothing but the color developing technique. By using Nestler's tube, the color depending upon the color intensity, we have to find out the concentration of the sample. So this is similar method with the Nestlerization, but it is a quite convenient than the particular Nessler's method. So it is less affected by the turbidity of the sample. If any kind of turbidity is present in the sample, it is not much affected by the particular method. So the more specific than most of other methods. It is specific in nature. We have to find out specifically the protein. So there is no any kind of interferences occurred, such as the carbohydrates and other kind of thing, which we have already seen in the previous some methods, that there is some interferences occurred due to the other methods. So it is relatively simple and can be done within a small period of time, that is around 1 to 1.5 hours is required for completion of the Lowry. Completion of the analysis of protein by Lowry method. Now what are the disadvantages? So generally what happens, whenever we are considering the different kinds of proteins present in the sample, so every protein shows different kind of color, and hence the color varies with the different proteins to the greater extent than the Biuret method, and hence we cannot properly identify, or we cannot properly quantify the given protein content. So color is not strictly proportional to the protein concentration. Then the reaction is interfered with the varying degree of sucrose, lipid, phosphate buffers and monosaccharide and hexoamines. So these are the interfering substances are present, which will cause some kind of interferences in the analysis. High concentration of the reducing sugar, ammonium sulfate and sulfhydryl compounds interfere with the reaction. So these are the disadvantages of this method. Now the next one is the dye binding method. So dye binding method means what? Whenever we have to analyze the particular substance, we have used some kind of dye, and dye having a role to show the color, okay, and depending upon the binding tendency, that it means that how much amount of the particular protein is absorbed, it is exactly calculated by using the spectrophotometric method whenever we have using the dye binding methods. So here first we will use the anionic dye binding method. So the principle, so the protein containing sample is mixed with known excess amount of the anionic dye and in a buffered solution. So the protein bind the dye to form an insoluble complex, and the unbound soluble dye is measured after the equilibration of the reaction and the renewal of the insoluble complex of the centrifugation or the filtration. So look at what happened. If you consider the particular, if you take the particular protein sample, into that we have add excess dye, then protein forms a insoluble complex with the dye, and there is a unbound or soluble dyes remain. The anionic sulfonic acid dye, including acid orange 12, orange G and amido black 10D, binds the cationic groups of the basic amino acid residues such as imidazole of histidine, guanidine of arginine and alpha amino group of the lysine, and the free amino terminal group of the proteins. So the amount of the unbound dye is inversely related to the protein content of the sample. So it means what happened? The bounded protein, that means excess of dye whenever we have added, so how much of dye is consumed, so accordingly the unbound dye proportion, it is inversely proportional to the protein content in the given sample. Now what is the procedure? So the sample is finely ground around a 60 mesh or smaller size of particular sample which we have to take, and then add a excess of dye solution with the known concentration. Okay, here we have add the dye in excess of known concentration. So the content is vigorously shaken, and to equilibrate the dye binding reactions and filter or centrifuge to remove the insoluble substances. Absorbance of the unbound dye solution in the filtrate or supernatant is measured, and the dye concentration is estimated from the dye standard curve. So here we have to plot a standard curve, and from that standard curve how much amount of unbound dye is remain, accordingly we have to find out how much of the protein is present in the given sample. So a straight calibration curve can be obtained by plotting the unbound dye concentration against the total nitrogen, which is determined by the Jadhav method of a given food covering a wide range of the protein content. Now the protein content of the unknown sample of the same food type can be estimated from the calibration curve or from the regression equation calculated by the least square method. That is, we have to use here some statistical parameter to get the exact concentration of the sample. Now what are the applications? So the anionic dye binding has been used to determine the proteins present in the milk, wheat flour, soy products and meats. Now what are the advantages? So this method, it is very rapid. So around 15 minutes or less time is required for completion of this analysis, and relatively accurate analysis for analyzing protein content in the various kinds of food products. It may be used to estimate the changes in available lysine content of the cereal products during processing, since dye does not bind altered unavailable lysine. So we have to estimate the changes in the lysine content during the various processing steps. In this method, we doesn't require any kind of corrosive reagent. So it doesn't measure the non-protein nitrogen. So we have to selectively measure the protein nitrogen only. So this method is more precise than the Jadhav method. Now what are the disadvantages? So this method is this method is not sensitive, and milligram quantities of the protein is required for the standardization and all that things. Protein differ in basic amino acid content and so differ in the dye binding capacity. Therefore, calibration curve for a given food commodity is required. So we have to specifically require the calibration curve for that particular food sample. So it is not suitable for hydrolyzed proteins due to binding to the N-terminal amino acids. Then some non-protein components bind a dye, that is a starch or protein, calcium or phosphate, and cause errors in the final results. Now the next one is the Bradford dye binding method. So this is another method in which we have to analyze the protein content. So the principle, when Coomassie brilliant blue, that is G50, binds to the protein, the dye changes color from reddish to bluish, and the absorption maximum of the dye is shifted from 465 to 595. The change in the absorbance at 595 is proportional to the protein concentration of the sample. So like other dye binding methods, the Bradford relies on the amphoteric nature of the proteins. So what is the amphoteric nature? So protein behaves as an acid as well as a base. That's why they behave as a amphoteric nature, and when the protein containing solution is acidified to pH less than its isoelectronic point of the proteins of our interest, the dye added binds electrostatically, and the binding efficiency is enhanced by the hydrophobic interactions of the dye molecule. In case of the Bradford method, the dye bound to protein has a change in the absorbance spectrum relative to the unbound dye. Then what is the procedure? So the Coomassie brilliant blue is dissolved in 95% ethanol and acidified with 85% phosphoric acid. Then the sample of the protein containing 1 to 100 microgram per ml concentration and the standard BSA solution are mixed with the Bradford reagent. So the absorbance at 595 is read against the reagent blank, and the protein concentration in the sample is estimated by plotting a BSA standard curve. Okay, similar manner we have to plot a BSA standard curve, and from that we can find out the amount of the proteins present in the sample by using the calibration curve. Now what are the applications? So the Bradford method has been used successfully to determine the protein content in Watts and Beer products and in potato tubers. This procedure has been improved to measure the microgram quantities of the protein. Then what are the advantages? So this method, it is rapid method. It can be completed within 2 minutes. This method having the results which are reproducible. The results which are getting are reproducible in nature. Means what? There is a reproducibility in the results. We have to reproducible that results by some means. Then sensitive. It is very sensitive, several fold more sensitive than the Lowry method. So several times it is more sensitive than the Lowry method. No interferences from ammonium sulfate, polyphenols, carbohydrate such as sucrose or cations such as potassium, sodium and magnesium. It measures proteins or peptides with molecular mass approximately are equal to greater than 4000 Dalton. So these are the advantages. Now what are the disadvantages? So it interfered with by both non-ionic and ionic detergents such as the Triton X100, sodium dodecyl sulfate, that is SDS. However, errors due to small amounts of, that is 0.1% of these detergents can be corrected using the proper controls. The protein complex can bind to quartz cuvettes and the analyst must use glass or plastic cuvette. That this is one of the disadvantage, that the protein complex can also bind with the quartz material. Then color varies with the different types of the protein and the standard protein must be selected carefully. So these are the disadvantages of the method. So this is all about the lecture. In the next lecture the B we will see the UV visible absorption method for the estimation of the protein. Thank you.